Okay, so let's go ahead and get to it. First, we're going to talk about what genetics is and who Gregor Mendel is. So before we get to Gregor Mendel, what we're going to talk about are some common misconceptions before Mendel came along. And so people did not have a means for understanding how traits are inherited. So these are several of the examples of like how people put together traits were inherited. So some people thought sperm contained little people, which is called a homunculus. They even have a drawing in some old text. There's like a little man in there. Some other beliefs are like begets like, so offspring are like their parents, but that doesn't really say how. This one, females control the traits, males control all the traits. There's a blending of the two. Inheritance of acquired characteristics, so like the giraffe's neck, like the famous example, like the giraffe stretched its neck and it passed that trait on, which is not true. And then the idea that particles called gemmules carry traits that we inherit. So this one's more along the lines of the correct answer. These ones are just kind of ridiculous sounding. So let's get to Gregor Mendel. So a man before his time, or a man, I guess ahead of his time is what I should say. So Gregor Mendel, who he was an Austrian monk from a very, very poor family who went off to the university to learn. And while he learned, he worked on farming. Since he came from a farm family, he helped like grow the food at the university that he went to school for, and he paid for his education that way. So very poor, came from a poor family. He went and he studied like mathematics, physics, biology. I think he even studied like philosophy at the university, and he didn't want to go back to work on his farm, but he wanted to be a monk, which at the time was like very highly held like career choice. So he went to an abbey in Austria, and he still took a liking to growing and farming plants. And so he grew these pea plants, which produce a lot of peas in a short amount of time. So they were a good food source, and they're pretty cheap and cheap to keep. So while he was growing these peas, he started to notice like different characteristics of the peas. There were tall pea plants, there were short pea plants, there were purple pea plants with purple flowers, pea plants with white flowers, pea plants with green pods, yellow pods, green seeds, yellow seeds, round seeds, wrinkled seeds, like all of the above. And so what he began to notice is that some of these traits showed up more than others. And so he started to do some experimenting while he worked in the abbey. And so why did he choose peas? So again, peas were just available to him. But what he didn't know are some of these things. Many of the traits that he talked about, so pods, seeds, flower color, height of the plant, they were There were traits in which it was either one or the other. So they only had two different choices. So it was like the flower color was either going to be purple or yellow. That was its only choice. The seed color was only going to be green or yellow. That's its only two choices. So it's either going to be one of the two. So many simple traits. So they're a very good plant to study. And actually, we still use the pea plant in botany studies. So it's pretty cool. Many of the traits were true breeding. So we'll talk about that in a second. The pea plants were really easy to manipulate. So once he began experimenting on them, he could easily manipulate a male and a female together. So they were they're what we call monoecious. And monoecious means that there is male and female parts on one organism. So plants are typically monoecious. There's male, female flower parts, right? There's like the pistil and the stamen, and then there's the ovary. So it was really easy to like pull the pollen off and pull the ovary off and what have you, and use that in his experiments. Each pea that was created was a new individual. So once he crossed some of the pea plants and they started to produce pods, each of these peas is a new plant, right? So he could plant them and see what they grew into and then what traits that they exhibited. And there were short generation times, so he didn't have to wait years upon years to see the new babies sprout and see what traits they had. He could grow them like within a season and then see what he got. And all he had was time. All he did was work at the monastery in the abbey. So he had lots of time to study his peas on downtime because he didn't have a family. So pea traits, right? I like this meme up here in the corner. Gregor Mendel, the father of genetics, likes pea plants over other plants, but very wise choice in his part, and kind of like a serendipity moment there. So, oh, my pen is dying. Hopefully I can get it fixed for later. So Mendel, when he began writing his experiment, he began writing like his materials and methods and his data and everything. He used different notations, and these are some of the notations that we still use. So P means parent generation. F1, F2 means filial generations, and it's like what number. So the parent generation is going to produce F1, and then the F1 generation makes F2, and then F2 will produce 3. F3 produces the F4 generation, the F4s create the F5 generations, so on and so forth. So there are some other terms. So self-cross means a plant that can cross with itself. So the male parts of the pollen would be used to fertilize the same plant. And then true breeding would be when parents have the same traits that they always pass on. So if it was a purple tall pea plant, it would always pass on those purple traits and the tall traits to its offspring. Okay, so just some vocabulary. There is a lot more vocabulary coming, but I just wanted to kind of give you some of it right now, because we are going to go over a quick cross. Okay. Are there any questions so far before I move on to talking about crosses? I'm hoping most of you either began reading or finished your reading in the textbook, that you kind of have an idea of what's going on. All right, so let's just look at the pictures for right now, and then we'll add in the letters later. So, P generation, to give you a visual. The P generation is this, so we can say that's the we say female, and this can be the male of the parent generation. So parent generation is going to produce the F1 generation. Okay. Well, I wish it didn't erase everything. I hit the erase button. All right, there we go. So female, male. So the parent generation, the P generation, male, female, male parts crossed with female parts, are going to create the F1 generation. The F1 generation is going to produce the F2 generation down here. So what we're seeing here is that use of one of our vocab terms, true breeding. So these parents, after crossing them once and then crossing their offspring once, we see that they are true breeding, so that every offspring from these parents are going to always produce that purple flower color. Now, if there was a white flower that showed up like here or here, that would mean that these are not true breeding. So true breeding means that they always give off that purple trait. So what could be the alternative? So the P trait, let's see. I want to make sure I'm not getting ahead of myself. All right, cool. So Mendel crossed different plants. So he'd crossed the purple plant, which we just saw, right? So purple with purple, we're always going to get purple. But once he crossed purple with white, he got purple, and then he got white again. So if we're thinking about, like, the parents are a blending of the two, or the traits always come from the female, or the traits always come from the male, this just went against everything that geneticists, well, they didn't call themselves that at the time, but everything that geneticists up until the mid-1800s knew about traits. So he was really confused when this white flower came up, because if a white flower came up, he thought it would come up here and not down here. So again, let's just go back over it. So purple true breeding, so meaning it's always going to give off purple. True breeding white means it's always going to give off white. So if we crossed white with the true breeding white, we'd always get white. So together, we got purple and purple, right? True breeding purple, so we got all purple. But then he crossed these with each other, or he self-pollinated this flower, meaning the pollen pollinated the ovary on the same flower. He got purple, 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 and white. So 25% of the offspring were white. So he thought that there must be something there in these plants that's causing this white trait to be masked or covered up, because it's showing up later on with subsequent crosses. So let's look at some more terms. Actually, let's see. We're gonna let's go to here first. So if we're talking about that purple flower, right, true breeding purple flower with a, you won't see my white, so true breeding white flower. What he ended up coming up with is that there must be a couple of traits that give rise to what we're seeing, right? So those traits, I'm going to go back to my vocabulary. So those traits that he was seeing are called the phenotype, right? So that's like purple, white, purple, white, green seeds, yellow seeds, tall versus short. So the phenotype is always what we're going to see. And now the phenotype is controlled by what is recorded in your genes, right? We've already discussed many times already that your DNA controls how your body is formed, right? Ever since day one, sperm meets egg, you were formed into a diploid organism. From that point, from that single cell point, every cell in the body, or in this case the plant, has the same DNA. And so that DNA is going to code for traits, right? So your DNA, your genotype in this case, is coding for these traits. So Mendel decided that he had 2 copies of every trait, or he had pairs of traits. And so he called these a genotype, or he actually called them like hereditary particles or gemules, but now we know them as genotype. So that's what codes for your traits. So you can be what's called like a homozygote or a heterozygote on any trait that is like a Mendelian trait. So we'll talk about that later. But so you can be a homozygote. So if you're homozygote, that means 2 of the same. So homo means same, hetero means different. So if you're a heterozygote, you have 2 different traits or 2 different alleles for that trait. And then if you're a homozygote, you have 2 of the same alleles for that trait. So now you might be asking, what's an allele? An allele is the hereditary particle. So it's like a segment of a gene. So if we say flower color, so flower color is the gene. The allele, oh my goodness, the allele for that could be either purple or white. Okay, so don't confuse gene and allele, they're different. So you have genes, you have a bunch of different sets of genes, and then you have variations of those genes, which we call alleles. So the gene that controls flower color, you can have an allele for purple or you can have an allele for white. That's the only 2 things that will fit in that spot. So same goes for seed color. The gene that controls the color of the seeds can either have a sequence that codes for green or codes for white. So alleles are different variations. So you have 2 slots, 1 on each chromosome that controls that. So alleles can be dominant, they can be recessive. So a dominant allele is going to mask recessive alleles. So it's always going to show up. We tend to, if you have a dominant allele, it's going to show up. We tend to name the dominant allele, we tend to like write the letter for the dominant allele as a capital. And then the recessive allele is just the lowercase form. We're not going to use W, right? So a little p would be white in this case. Big P is purple. Purple would be dominant. And then little p, the recessive, is just the other color. In this case, it's white. Okay, so here we go with our vocabulary. If we have a big P, purple, homozygote, so that means we have 2 of the same, with a little p, homozygote. This would be a dominant. This homozygous for dominant. Homozygous, dominant, and then homozygous recessive. So if we cross those, we get all heterozygotes. So what that means is that one allele is coming from dad, right? One allele is coming from mom, or maybe it's vice versa. So what did we just learn last week? Is that in meiosis, your genes get halved, right? So we start with a diploid germ cell, and we end up with haploid gametes. So the gametes that are passed on, what are we giving off? We're giving off a set of genes that's only 1 through 23, so it's only a half a set. So in this case, it's either going to be a big P or it's going to be a big P that's giving off. So there's no real variation here. And then the same thing for this one. It's either going to be a little p or a little p that's going to give off, right? So what I'm saying is that where these traits are located, in this case, for pea plants, it's on number 4, chromosome number 4. So they're going to either give off this one, which looks like could be tall, and then they could also give off this one, which would also be good. They could give off that wrinkled, and then we have little W, which is smooth. So remember, at the end of meiosis, we're only giving off half a set of chromosomes. So we're only giving one copy of each of these. And it's totally random which copy we're giving off, right? Because of random alignment at metaphase 1 and metaphase 2, random assortment. And then it's totally random, like what sperm or egg gets fertilized or takes part in fertilization. So it's totally random which one we get. And it's like a 50-50 chance. So you can think of it as like flipping a coin. So it's going to be like heads or tails, which one we get. So I kind of skipped ahead, so don't look at that. Mendel's laws. So he has 2 laws. One law is the law of segregation. Maybe I should play this. Law of segregation. And what the law of segregation means is that each allele is, I don't want to use the term segregated, it's sorted. Each allele is sorted into its own gametes. So each allele for that trait. So if we have a purple plant that is maybe like a heterozygote. So big P, little p, that big P can be segregated into one, and little p can be segregated into a separate one. So it's either going to be big P 50% of the time, or it's going to be little p 50% of the time. There's also another law which talks about like random alignment, which we've already covered, or independent assortment, which just means that each gene doesn't, let me type it here, independent assortment. Each gene does not affect others. Meaning like flower color, if you have a purple flower, that doesn't mean anything else for the plant. It could be tall, it could be short, it could have wrinkled seeds, it could have smooth seeds. So each gene does not affect other genes. If it did, that would be called like epistasis, or it could be called like multiple alleles, which we'll talk about later on in the chapter. But for our sake, for Mendel's laws, he has which one of these is that genes don't affect other genes. But of course, like I said, there's always exceptions to that rule. Now, I know we've already talked about law of segregation, so I think you understand what that means. Independent assortment. I think you understand what that means too. Pretty straightforward stuff. So let's talk about some of these real quick. I'm just going to fill in 2 of these examples. It should be pretty easy, right? Big A would be a homozygous trait. Big E, little e would be a heterozygous trait, right? So if we're talking about law of segregation, the parent could give off this big E, or the parent could give off that little e in that case. And then one more. So a little f and a little f would be homozygous for that trait. So we can add to these and say that this is homozygous dominant, this is heterozygous, and then this is homozygous recessive. So we typically don't say anything after heterozygous. So when we say something's heterozygous, you just automatically know that it has one of each, right? But when we say homozygous, you don't necessarily know if it's homozygous dominant, does it have 2 dominant traits, or does it have 2 recessive traits, in which case it would be homozygous recessive. So we have to decipher that on the homozygous, but heterozygous, we don't need to say it because it is what it is, right? So purple flowers dominant to white. I'm not sure what phenotypes. Oh, this would be purple here. This would be purple again, and then down here would be recessive, so it's white. Okay. So these are just some of the examples from the worksheets I put up on Moodle. So if you want to practice more on your own with them, you can. And then if you want to send me like your answers, I can check them and we can go over them if you'd like. Are there any questions about homozygous, heterozygous, and then like genotype and phenotype before we move on? That was an activity I did in class. All right. So setting up a Punnett square. So let's say the question's asking you to set up a Punnett square and you're crossing a true breeding purple plant, which means you have big P, right? Homozygous dominant. True breeding means it's always going to give off whatever trait comes after the word true breeding. So if it's a true breeding purple plant, it's always going to give off purple alleles. Right? And then we're going to cross our true breeding purple with our true breeding white, so that we're always going to give off the white allele in that case. So if we set it up, big P goes up on top of our Punnett square. So you just kind of draw a window frame, right? And then little p goes on the side. It really doesn't matter if you put little p up here or big P over here. You just have to be on the same side. So if this is parent number 1, they have all the alleles from parent number 1 have to be here, and then parent 2 would have to be there. Okay. So we're going to cross them. We take 1 from the top and 1 from the side, right? So how did I get that? So big P is here in this column. It's going to go here. Little p is on this row. So it's going to fill in there. So if we fill in the column, big P would be there. And then if we go to the side, little p is on that row, so we fill it in there. And we typically write the dominant trait first, just because that's like how the English language works. We put a capital letter before a lowercase letter So that's the reason why we write it that way. There's not really a true reason. It's just kind of how etiquette is now. So here our P generation, right? P generation produced our F1. Right? So our filial generation, our first generation offspring are here. So now we've produced these guys. We're going to cross them again. So I'm going to change my color. Our F2s are big P, little p, big P, little p. So what are these? These are heterozygotes. Heterozygotes, which means they're going to give off a purple and a white trait. So if we take parent 1, so big P, little p, and then parent 2 is on the side, big P, little p, and then we cross them. So big P at the top comes down, and then big P on the side comes over. We have 2 big P's there, and then the same thing. So little p comes down, big P from the side. And then we go to the next row. Big P up here comes down, little p comes over, and then little p comes down, little p comes over. So what we end up creating is a chance of 75% purple, and then 25% white. Right? This is our white. So I hope this helps you realize that the only way to get a white plant or a homozygous recessive is that it has to be homozygous recessive. If we get a plant that has a heterozygous genotype, its phenotype is going to be the dominant trait, right? Dominant trait, the capital letter, the genotype represented by the capital letter, is always going to mask over that recessive gene or that recessive allele. So even though all of these F1s are big P, little p, they're only ever going to show that dominant trait. They are going to what's called carry that white gene, and that's why it comes up here. So remember, big P is going to give off 50% of the time, and so is little p in meiosis. And because we can give off each of these alleles equally the same as like flipping a coin, heads or tails, we are still going to give that white gene. And so we are going to have eventually white offspring if we cross 2 heterozygotes. So even though we have the gene here, we're not necessarily going to have the white characteristic or the white phenotype. So genotype is going to be different almost, well, a lot of the time. Genotype is going to be different from the phenotype a lot of the time. So even though something looks purple, that doesn't mean it's homozygous dominant. Okay. Just keep that in mind for some of the questions that you're going to see on the quiz later in the week. So these are some practice questions I pulled from the worksheets on Moodle. So let's try to complete like 1 or 2 of these. We got 7 minutes. We can probably do all of them. Let's try to complete some of these. So let's draw a Punnett square. So there's our square. All right. Question is, complete a Punnett square and determine the probability of genotype and phenotype. So phenotypes here. All right. So we have a true breeding round crossed with a true breeding wrinkled, and in our directions, read that part, round seeds are dominant. So that means we will have big R for our true breeding round. Why do I know that it's big R and not big R little r? Because it's true breeding. So remember, true breeding always going to give off that allele or whatever trait follows that true breeding word. So true breeding wrinkled is always going to give off those alleles for wrinkled. Always. Now, if you notice down here, these do not say true breeding, so we're going to have to figure out what they are. All right. Parent 1 goes up top, parent 2 goes on the side. Now, if we fill them in, so big R comes down, little r comes over, so on and so forth, we'll find out that every square comes out the same. So all of our genotypes are going to be big R, little r, or we could just say heterozygous. Okay. So what does that mean? Okay, cool. Like they have one dominant, one recessive allele on their chromosome. But what does that mean that we'll see? That means that we're going to have round peas. So all the plants that are produced from these parents will exhibit round peas. Okay, because that dominant trait always masks that recessive trait. Questions about that one before we move on? If it asks you for a ratio, I don't know really what the ratio would be for this one. I guess it would just be 100, or maybe it would be like 1 to 0, but I don't know. Don't quote me on that. 100%. All right. Let's see. I'm going to use a different color for this one. Keep it Halloween themed. Heterozygous with a homozygous wrinkled. So heterozygous, even though it just says heterozygous, we already know it's one of each, right? Two different is what hetero means. Homozygous wrinkled, so we have to go up here and read again. Round seeds are dominant over wrinkled seeds. So that means that wrinkled seeds must be our recessive. So that is what that parent has. So parent 1, 50% of the time can give off big R, 50% of the time gives off little r. Parent 2 gives off little r and little r. So if we cross them, we're going to get something different than before. What we see is that we have a genotype that is big R, little r, and little r. And then our phenotypes, we're going to have round and wrinkled peas. So if we need a ratio, this ratio would be 1 to 1. So there's one round plant to every one wrinkled plant. You could say 2 to 2, but you know how math is. They always want you to simplify your answer, so that's why it's 1 to 1. All right, let's do the last one. My squares keep getting bigger. Homozygous round. So homozygous meaning we have 2 of the same allele. Round, we know that it is dominant, so it's capital R. And then we're crossing it with a heterozygote, which means it has 2 different. Right. Parent 1 gives off big R 50% of the time, gives off its other big R 50% of the time. Parent 2 gives off a big R 50% of the time and gives off a little r 50% of the time. So if we cross these, what we end up getting, I haven't set myself up yet, phenotypes and phenotypes. We have big R, big R, big R, little r, big R. And then our phenotype, so big R is going to be round, and then big R, little r is also going to be round. So 100% round in this case. So even though we have two different genotypes, they're both going to be round. So in this case, like if we took these two parents and we weren't sure if the offspring were if they were like true breeding, so notice it doesn't say true breeding, right? So let's say we didn't know, and we just crossed these two parents that were exhibiting round teeth. We crossed them and they gave offspring like this, we would have to cross them again just to be sure. So if we cross a heterozygote like this, what we would end up getting is a mixture, and we would get wrinkled seeds eventually. And then that's how we would know that these guys weren't exactly true breeding. There we go. Let's say red, and let's say that one's going to be black. So you have a big P, little p. In this case, I colored them differently to represent two different alleles, and this would be a genotype. So that genotype is what's recorded on your chromosomes. The phenotype would be purple, right? So it's what we see. So the genotype codes for the phenotype. Now we have one big P, one little p. In the case of complete dominance, which is all we've learned so far, is that the dominant trait is going to always mask that recessive trait. So the white isn't necessarily going to show up in the phenotype of the plant. However, this plant can be a carrier of the white gene or the white allele. So let's see what that means. If we take big P, cross it with little p. What does that mean? This is big P is homozygous dominant. Dominant. Little p, homozygous recessive. So these are also going to be referred to as true breeding, meaning they are always going to give that allele or that trait, or that purple trait or that white trait. So if we think about it in terms of meiosis, big P, oh, let's see. I wish I could just erase this one part. Let's just do it over here. So this parent, the chromosomes that they have, so let's say we're talking about chromosome number 3, right? Chromosome number 3. They're always going to give a big P. And I know you can't really tell those are big P's, but Anyway, they're always going to give off a big P, so their four gametes that they create at the end of meiosis 2 are always going to have that big P. Okay. So if we cross a true breeding homozygous dominant with a true breeding homozygous recessive, we can do the, oh, if I can draw, here we go. So we can do the Punnett square, and what's going to happen, well, first how we set it up is 50% of the time, the first parent, the homozygous dominant parent, is going to give a big P, right? So that comes there. 50% of the time, they're also going to give a big P, so that's their only choice. And then same with the other parent, so little p, little p. So we split them up because after meiosis, we only have half of our genes, right? The one parent can only give half of their genes, that's why they can't give 2. That's why we don't put 2 here, we only put 1, right? So it's half. Right? 1 is half of 2. Okay. So when we cross them, big P comes down, little p comes over. So we get one set of genes from each parent, correct? So this is like fertilization. So this big P came from this parent, this little p came from this parent. So one set from each parent. We end up with a probability of heterozygotes at 100%. Okay, so these heterozygotes, you might also see them called a hybrid. You might see them called a hybrid, meaning that they are not true breeding, and they have a mixture of the two alleles. So if we, let's see, oh, let me backpedal real quick. What I described over here on the right side is that first law we talked about yesterday. It's the law of segregation, meaning that one allele is given to each offspring, or one set of chromosomes, or one set of genes is given to each offspring. Okay. So, moving on, if we cross, let me see, I keep using red. So if we cross two hybrids, big P, little p, with big P, little p, what we end up getting is something totally different. So this would be, let me use the color black for a square. How would we set this up? So half and half, and then half and half, right? Big P, little p. So 50% of the time this parent will give the dominant allele, 50% of the time the parent will give the recessive allele, so half of 2, right? And then same thing for the other parent, big P, little p. So when we cross these, we get a mixture. When we cross hybrids, we get a mixture of traits, which is pretty cool. Genetic variation is always a good thing for survival of the population. So we cross those. What we end up getting is a very different setup than we did prior. So big P, well, let's see. So the phenotype and the phenotypes here are going to be different. Phenotype here is going to be 3 to 1. 3 purples to 1 white. Or we can think of it as 75% chance of having an offspring that's purple, 25% chance of having an offspring that is white. And the genotype is different. It is 1 to 2. to 1, meaning 1 is going to be homozygous dominant, 50% chance, or 2 having heterozygous genes, and then 25% chance of having an offspring with homozygous recessive genes, phenotype. So, oh, we didn't talk about, these would be purple. Okay, that would be the phenotype, right? All right, so let's put some notations on these. I'll use green for this. All right, these here are what we called the parent generation. The parent generation, when we cross them, we get an F1 generation. That F1, if we cross these, which sounds like incest, we would get these guys here, which is an F2 generation. So some of your quiz type questions that you might see will give you the parent generation. So they'll call them the P generation. And then they will ask about not the F1s, but they'll ask about the F2s. So it's your job to know that you have to do 2 crosses for problems that ask those questions. So if they give you the P generation, you'll have to cross the F1s, get the F1s, cross them again to get the F2s. Okay, so dihybrid cross. What is it first? So dihybrid cross is showing the crossing of 2 traits which are located on separate chromosomes. So let's think of it in terms of meiosis. Okay, so we have 2 pairs of chromosomes here. This could be like chromosome number 1. This could be like chromosome number 2. And on these chromosomes are the genes for these traits here. So here's the gene for that trait. And then here's the gene for that trait on that chromosome. So on the first chromosome here, so chromosome number 1, the first of the pair, could code for, let's say, a big Y. This one would code for a little y. And then down here we could have a big R and a little r. So after meiosis, so let's see, this would be what phase of meiosis? Phase 1. Okay. So at metaphase 1, remember we have homologous pairs of chromosomes line up. So 1 and 1 are next to each other, 2 and 2 are next to each other, 3 and 3 are next to each other. And when we switch into anaphase 1, those pairs get pulled to the sides of the cell. So that would create 2 new cells with different genes. So that's one combination that we could have, right? So we could have, well, technically after another division, we'll have 2 of each. But anyway, so one combination of these genes is to have a big Y and a big R in one, and a little y and a little r in one. We could flip it, and we could put, remember, we have one source of genetic variation is how these chromosomes line up. So if we flipped it and we put use a different color, maybe instead we had big R on this side and little r on that side, which would cause a different variation of chromosomes or different gametes, right? So based on how they align is that combination. So what we put on our dihybrid cross is all the combinations that we could have with those alleles that we're given. So we tend to use like the FOIL method so that you don't have to write all this out every time. It would be like this. So first would be big H, big S, and then outside would be big H, little s, and then inside would be big H, big S, and then last would be big H, little h, little s. So remember that how you set up your Punnett square is by having the number of chromosomes or having the number of genes that you're given. So since we have four genes, or we have 4 alleles by these parents, we would be putting 2. We would be putting 2 alleles above each box. That, and then for the other parent, they don't have any variation. Oops, put an extra row. Oh well. So cross that out. What we end up getting, big H, big S, little s, and that's the same all the way down. Big H, little s, little s, and that's the same all the way down. Little h, big S, little s, and that's the same. And then little, and that's the same for all of those boxes. So what we're showing you here with these dihybrid crosses is that the genes are not influenced by each other. So remember, I showed you these chromosomes, which contained, we'll say, these, we'll use H and S since we're on this slide. But these chromosomes have genes on them that are separate from each other, or the chromosomes are separate from each other, and so each gene is not influenced by each other. Meaning that in this case, hairless or hair or stripes or no stripes, we can have hair and we can have stripes, right? Hair with stripes. We can have hair and no stripes. We can have no hair and stripes. And we can have no hair and no stripes. So the alternative to this is that we would have linked genes. So linked genes are genes that we typically see together. So think of somebody that you've seen, or maybe you even, you have red hair. You have freckles. Red hair and freckles are 2 different genes, but they're linked because they're on the same chromosome. These genes, hair and stripes, these genes are on separate chromosomes, and so they can be inherited independently of each other. So we aren't going to always see a combination of the two. So we're not always going to see, well, if you have stripes, you must have hair. That's not the case here. So they're independent. So that is Mendel's second law, which is the law of segregation. So we can always have a combination. Read this right here. So segregation and recombination of alleles for one trait has no effect on the segregation and recombination of alleles for other traits. So just because you inherit one trait doesn't mean you're going to inherit another trait. And that's for Mendelian genetics. Of course, the red hair and freckles is one of those exceptions to Mendelian genetics. We're going to talk about that probably next week when we talk about special inheritance. So, if you want me to go back, I know I moved on really quickly. So what we're showing here, this is the image from the textbook, is it's showing you that we can have peas that are yellow and round, we can have peas that are green and round, we can have peas that are yellow and wrinkled, we can have peas that are green and wrinkled. If this was not the case, we would have peas, we would have like 50% like this, and we would have like 50% like that if these genes did not segregate. But since they can segregate, we get a wide variety. Variety is good. Okay, so let's do this bunny question quick. So this bunny, rabbits have, let's see, two traits involving hair and ears. Okay, rabbit. So black hair is dominant to brown, and long straight ears are dominant to floppy ears. So ears that would stand up versus ears that flop down. So this guy, what do you think his genotype would be? What do you think his genotype would be? So if black hair is dominant to brown, straight ears are dominant to floppy ears. He would be little b, little e, right? So big B pertains to our dominant trait, so big B is going to be black. Long straight ears, big E in this case will be long, and then little e is floppy. So since this rabbit is exhibiting both of those recessive traits, in order to exhibit a recessive trait, they have to be homozygous recessive, right? So let's turn the tables. If this bunny was black, if it had black fur and floppy ears, we don't know if it would be big B, or big B, little b. So it's hard to tell with the dominant trait, but it's always pretty easy to tell with the recessive traits. So when something's exhibiting or showing phenotypically a recessive trait, we know for sure it has to be homozygous recessive. Okay. So let's get to the question. How about that? A male rabbit with the genotype big B, little e. So what is that? That's black and floppy. Black hair, floppy ears. It's crossed with a female rabbit that is brown with straight ears. Brown and straight. So we've set up the Punnett square already. We're going to fill it in. So the way we fill these in, we put the dominant first, followed by the recessive. So remember, four alleles here, you only put 2 above each one. And each one is representing a combination that we could get. So this one's not really showing the combinations well. This one is because we have that E, the heterozygote E. So if we fill all these in, what we end up getting is that, and that. So why didn't I fill in the rest? Because every column here would be the same. So all of these would be the same all the way across. We would have four like the first one, 4 like this one, 4 like this one, 4 like that one. All right, so how many of the 16 potential offspring or potential variations have long ears? Let's look at our key again. So long ears has to have a big E. So they could be homozygous dominant for long, or they could be heterozygous and show that long since it's the dominant trait. Dominant masks the recessive. So how many of the 16 have long ears? We have what? 8, right? So that would be these and these. So that would be like 50% of the time the offspring of these 2 rabbits will have, black hair and floppy ears. Oops. Oh no. I messed that up. Okay, let's fill these back in real quick. It always pays to go back and read the question. Okay. So black hair and long ears. So black hair, big B, long ears, big E. That one. And, well, I guess we have the same one. But anyway, 8 and 50%. How many of the 16 have black hair and floppy ears? So that would be the other 8, right? So 8 for these here. And then how many of the 16 have brown hair and long ears? None. Actually, none of ours will have brown hair. And how many will have brown hair and fluffy ears? So none. So strange, right? The female rabbit had brown hair, but none of her offspring will because the male rabbit was homozygous dominant. So every allele that this male rabbit can give off is going to be for black hair. The pedigree, you've probably seen them before. It's a chart that shows the relationship between people, and the lines are very important in what direction they are running. So if we look at this main graphic right here, just showing you examples, a horizontal line means that they married or that they had children together at least. And then a vertical line means the descendants of that coupling. Okay, so vertical line leading to 1, 2, 3. So the order 1, 2, 3 matters as well. The the offspring furthest on the left is number 1. That would be the first offspring, in this case, the oldest. And then 2, and then 3, the youngest being on the right. Now, if one of those offspring has a relationship and they have offspring themselves, I guess, what they would have is this other horizontal line leading to an individual who's not connected anywhere else. So that means that there is no relationship, which is a good thing, because that would be incest, right? So we want somebody that's not in the in the pedigree already. So then when they have children, it would be another vertical line. In this case, we're showing you identical twins. And so this is the shape that we would make for identical twins. So, yeah. Circles and squares mean something. Circles are females, squares are males. Kind of easy to remember. Square is male. Squares, males, I don't know. The darkened is somebody who has a gene. So what we make a pedigree for is to track genetic disabilities, disorders. Most of the time it's tracking like hemophilia or color blindness. So you need to create a pedigree to see a relationship and who might have it, who might be a carrier, so on and so forth. So the darkened color, or the darkened squares or circles, are going to be the individuals that have that gene, or whatever gene that we're tracking in this case. We can only track one at one time throughout a pedigree. If we track more than one, it starts getting really hairy. So we try to only to track one. It is easy to figure out who is a carrier. So if this is just a regular gene, so we'll say like brown eyes or something, or no, we'll say blue eyes. So let's say that this male up here has blue eyes, and obviously this female has blue eyes if it's darkened. So in order to get a homozygous recessive gene, we would have to have an allele come from both parents, right? So this mother must be heterozygote. And you can do the Punnett square to figure it out if you want. Let's do dad on top, mom's on the side. So big B, little b, little b, little b. So the other two in this case would be big B, little b, correct? Just by looking at the Punnett square. So we can do a Punnett square for the other instance if mom was homozygous dominant. Dad, mom's alleles go here. Remember, you can only give one, so that's why we put one on each box. So all of the offspring in this instance would be heterozygotes, and therefore none of their children would have blue eyes. Right? It'd be little b all the way around. So every instance would be big B, little b, meaning all their children would have brown eyes. But in this case, that's not true. So we know for sure that it is like this. Okay. So if we can go further, since we know that this male is big B, little b, this female, we don't have enough information. She could be big B, little b, or big B, big B. The only way you can tell for sure if somebody is a heterozygote or is homozygous is if you have a mixture of the two. If you have just one instance of it showing up, you can kind of lean one way. You can lean towards like, oh, it must be homozygous dominant, but there's really not enough information to discern that that statement. Okay. So let's look at an example from the text. Okay, sorry. So first generation, second generation, so we can see the different generations working down. What we need to figure out is the genotypes of these people, right? So we don't know. We don't know this one either. And so let's look at the genotypes that we do know, and hopefully we can kind of figure it out. Okay, so if the parents here, so dad, homozygous recessive, mom, heterozygote, each of their children have this. So if we do the Punnett square really quick, monohybrid cross, right? Dad, 50%, 50%, 50%, 50% for mom. All of the children are either going to be a heterozygote or, oops, that's not right, or homozygous recessive. So if we know that this individual is exhibiting the trait, so they're exhibiting that dominant trait, the only combination of alleles that we could have for that would be big A, little a, right? On the Punnett square. So we can also do the Punnett square for this over here. This one's a little bit harder to figure out, however. So let me show you what I mean. Set it up. Mom and dad are both a heterozygote. Maybe they did some genetic testing. Maybe they did 23andMe or Ancestry. So all of their offspring look like this, or they have the chance to create these offspring. So they have a 50% chance of creating a heterozygote, and then they have a 25% chance of creating a homozygous recessive, and another 25% chance of a homozygous dominant. So in terms of statistics, this is probably a heterozygote, but we don't know for sure until this individual has maybe some offspring of their own. And then number 3 here, the only way to get that offspring with a little a is this one must be this, or maybe they are little a, but let's look at the colors. Is this individual darkened? No, they're not, right? They're not the affected individual. So that means that they're not homozygous recessive, right? So they must be heterozygous. And you can do the Punnett square again to figure it out, but it'll pretty much look like this. So they had a 50% chance if mom, little a, dad, big A, little a, 50% chance of creating that offspring. Okay. Are there any questions about pedigrees? I'll pause in case somebody has a question. All right, doesn't look like it. Okay, so there are some examples here. We're not going to work them out though, since we're kind of low on time. But if you want to, after class ends, we could work them out together with our extra 10 minutes that we have. So let's keep rolling here. All right, let's talk about some non-Mendelian genetics. So remember, genetics are the ones that we've covered so far are complete dominance. So there's actually a Mendel law of complete dominance. that means that one allele is completely dominant over another. So a dominant allele is going to completely mask the phenotype of a recessive allele. So that's not the case all the time. We do have instances of incomplete dominance, which are shown here, and we have several more that I'm going to show you. So let's start with incomplete dominance. Incomplete is a blending of two dominant phenotypes. So the offspring are not going to look like the parents at all. Okay, so if we cross a true breeding red flower, so that would have to be homozygous dominant, right? If it's true breeding, that means it's going to give, true breeding means it's going to give that allele for red every time. And then if we cross it with a with a true breeding white flowered plant, in this case we don't use the same letter. It's incomplete dominant. So both alleles are actually dominant, and they kind of have like a tug of war between the two, and they end up kind of just at a stalemate. And when we cross them, we end up with pink. We end up with pink offspring. So it's that intermediate form. So if we've set up the Punnett square for this one, let's see, red and red, white and white, all of the offspring will end up as capital R, capital W, which means pink. And then if we take those, if we take these F1s and we cross them to get F2s, we end up with a mixture. So like 25% of the offspring will be red, 50% will be pink, 25% will be white. And this is seen in snapdragons. It's common. It happens a lot. If you buy some snapdragons in the spring and you plant them and you allow them to flower, go to seed, and then flower again, you'll probably see different variations of the snapdragon. Pretty cool. Okay, so something similar to incomplete dominance is called codominance. And codominance is written kind of similarly. You can write it like they have the notations here, but I like to keep the same notations. So if we cross a red and a white, we end up with all heterozygotes. And the heterozygotes for codominance look different than the heterozygotes for incomplete dominance. So remember, incomplete dominance, we had the blending, so they had like an intermediate form. So incomplete dominance, intermediate form. Codominance, both of the alleles will show up in alternating patterns. A prime example here is a roan cow. So they'll have splotches of red and they'll have splotches of white. Black and white chickens are the same way. So if you have chickens at home, which some of you probably do, you might have a black and a white chicken, and the black and white chicken is a result of mixing a white chicken with a black chicken, which gets a mixture of the two feathers. So this Punnett square for these will look the same as incomplete dominant Punnett squares. You just have to read the question carefully to figure out if it's incomplete or if it's codominant. So we get RW all the way around. So remember, a capital letter means a dominant trait. We don't write either of these as a lowercase because they're not recessive, right? They're both dominant. They're codominant. So that's why we use two different letters, and they have to be capitalized. You could write them this way if you want, if it makes a little bit more sense. It's just a lot of letters for me to fit into a Punnett square, so that's why I don't do it. Either way is right still. All right, so let's move on. Let's talk about blood types. So multiple alleles is another type of non-Mendelian trait. Multiple alleles is referring to multiple variations of a gene. So remember, we've been talking about our chromosomes, right? And on our chromosomes, we only have one slot for an allele, okay? So that allele, so far, it only has been one or two things. It's been like black or blue eyes, or it's been red or white color, right? So we've only had two choices. Well, I'm come to tell you that there are instances where we have more than two choices, and that's blood typing. So your blood type still made up of two combinations, but we can have multiple combinations. And how we write it is with the I's and these upper and these superscript letters. So if you have, let's see, I have blood type A. So I might have a genotype that's IAI or IAIA. That A or that B is referring to the antigens that are on the surface of your blood cells. So those antigens might be antigen A and might be antigen B. And that's why we have that. In the case of O doesn't have any, and so that's why we don't have a letter here. So it's resembling the absence of the antigen. Okay. So if we cross some of these blood types, so let's say mom is a blood type A and dad is a blood type B. Okay. Well, I probably should have spaced them out a little bit more. Let's see. All right, so mom is an A, dad is a B. Let's say that they got like genetic testing done and they know for sure what genotype they have, right? If you're blood type A, blood type B, you could have one of two genotypes. So when we cross them, we get a lot of letters I don't like to write. Let's try to make sense of what we have. Okay. Looking at our Punnett square, these two individuals' offspring have the chance of having all of the different types of blood types. So 25% chance of having a child with AB, 25% of having A, 25% of having B, and then 25% of having O. So these AB blood types, this guy here, is a codominant trait. So we use those two capital letters. Notice that the other letters are capital, right? So capital A, capital B. We aren't using lowercase here, so we're using the notation for a dominant trait. So now that we have two dominant traits, they're both going to show up at the same time. In this case, it's codominant. I'm telling you it's codominant. You're not going to have to like figure out if it's codominant or incomplete dominant. That's not going to be something that you need to do. But you need to know that it is codominant. So your blood cells of your blood type AB will have A and B RH factors. I think I've been saying antigens, but RH factor is what it's called. So again, O would be the absence of them, and then if there's IAI, that would mean it would just show the I or the A trait. right, because it's the capital. And then the little lowercase I is the recessive, and so it would be masked. And then that person would only create those Rh factors on their blood cells. So they wouldn't create those at all because it's recessive. Okay. Alrighty, let's see. Questions so far on incomplete dominance, codominance, multiple alleles? Okay, there are some other examples. The book likes to use the rabbits. I can't relate to the rabbits. That's why I like to use blood typing. But rabbits are a great example as well. They do have multiple alleles for their coloration, for their fur. Brown, black, Himalayan, and albino. And you can see the notations what allele codes for what. And they have kind of like a push and a pull for their dominance over each other. So a big C would be dominant all the time. And then we have these recessive Cs, which would only show up in the case that they're homozygous recessive. But let's see. I don't think you're going to have to figure out the rabbit example on your quiz. I think you're going to be asked about your blood typing on your quiz, which is why I think I showed you blood type. Okay, there are some examples that I have here, and there, of course, there's worksheets on Moodle that go over blood, not blood typing, but they go over non-Mendelian genetics. So we can do those later, but I just want to go over all of the information first, and then we'll get to examples. Okay. Sex-linked traits. Let's see, what else do we have left? Lethal, alleles, and epistasis. All right, so sex-linked traits are linked to the 23rd pair of chromosomes. So that 23rd pair of chromosomes, if you're a female, you're lucky because you have 2 coding chromosomes. If you're a male, you're unlucky, and I'm sorry. But you have only 1 chromosome that codes for genes. The Y chromosome, I did not draw it to scale. The Y chromosome is like this big compared to the X chromosome. It's very tiny. There's only a few genes on it that actually code for anything. But the X chromosome is where it's at. So, 23rd pair, right? That's our sex chromosome. And on that 23rd pair are a few different genes. One of them is color blindness. So the X codes for either the dominant trait of regular vision or the non-dominant trait of color blindness. Is this the one I want? Yeah, I guess so. Okay, this is the better example. So if dad, let's see, the son of a woman who is a carrier of a recessive X-linked disorder. So let's first talk about what a carrier is. What does it mean to be a carrier? So her genotype, she's going to have a dominant trait that I'm sorry, my dogs are rowdy today. If you are a female who's a carrier, that means that you have a dominant trait, which is masking a recessive trait. That recessive trait is the disorder or the affected allele. So in this case, if mom is a carrier for color blindness, what would her genotype look like? Big X, and I like to put a C for that recessive trait. You could put a capital C on the big X, showing that dominant trait if you want. I just leave it off. But in this case, I'll leave it on for explaining it better. So, Dad has the dominant trait, so that's a capital C and a Y. So he's not colorblind. He can neither be a carrier for color blindness either. In order to be a carrier, you have to have two coding chromosomes. In this case, if he had an X here, he could do that, but then he wouldn't be male, would he? But so if he was, his only other choice would be to be X with a lowercase c. In that case, he would be color blindness. He doesn't have another chromosome here to mask that one, correct? So that his only choice is to go with that X. Okay, so if we cross these two, let's set up the Punnett square. X with a big C. I should probably move this down. There. So X with a big C, X with a tiny c. Oops, messing it up again. It's Friday, it's almost the weekend. All right, X, big C, X, little c. And then X with a big, and then Y. Right, so if we cross these two, what we end up getting are some pretty nice genetics. Big C, Y, X with a little c, and Y. All right. So their offspring, of course, 50% chance of having a female, 50% chance of having a male, right? We all know that. So half of their children are female. Those females that they will have as children would be either a unaffected daughter, meaning that she has two of the regular genes, two of those capital C's, or they would have a carrier daughter, in which case she would be like mom. She would carry for the gene, but she's not exhibiting the gene because she has a dominant trait that's masking it. On the other hand, their sons, so 50% chance of having a son, correct? They can have an unaffected son, which would be X, capital C, Y. So he would get that dominant trait from mom, that dominant X from mom, and then he gets Y from dad. Or he can get mom's other X, which is the bad X, in which case he would then be colorblind. So 25% chance of having a child that has that disorder. It's not really a disorder, but I guess that's just the medical terminology behind it. So you might see questions on the quiz that ask about like what percentage of these two people's offspring will have that sex-linked trait. So you need to know how to cross them and then how to look and decide if their child has the trait or if they're a carrier or so on. So males cannot be a carrier if either they have it or they don't, because they only have that one X. That Y doesn't do anything for color blindness traits in this case. Females can either be a carrier or they can have it if they have two X's with that bad trait, right? They can be big X with a little c twice. And they could be colorblind. It's pretty rare for a female to be colorblind. Or they can be totally fine and have capital C. So people always come to me and during this chapter and they're like, I thought girls couldn't be colorblind. They can, it's just rare. It can happen. Alright, so lethal alleles. What is a lethal allele? I'm not going to go super far into this. Lethal alleles are recessive, or they could be dominant alleles. Either way, the homozygote is what's going to create that lethality. So the textbook used this Manx cat as an example. All Manx cats have short tails. A normal house cat is just a normal house cat, but a Manx cat has, I guess this would be an incomplete dominant trait. So a capital M and a capital M with that L on it creates a shorter tail. So it's like a bobcat kind of. If one offspring, if we cross two of these Manx cats, we will get 25% genotypes, or 25% of their offspring would get this genotype. And that genotype causes malformation of the vertebrae, like throughout the entire cat, and not just in the tail. And that causes that offspring to die before they're even born. So lethal alleles are a thing. I don't know a lot of lethal alleles. And I think it's because that we don't know of a lot of the lethal alleles. I have Dachshunds. I have 3 Dachshunds, and I've bred them a few times. And there is a lethal allele in Dachshunds, which is called like a double dapple trait, and it causes like problems with their vision and their hearing, and so they could be deaf and blind, which is not a good thing, and it could cause them to die if they have really bad problems with their eyes. But with humans, I'm not sure. I'd have to look it up. I just think that we don't know a lot about lethal alleles because that offspring dies before it's even born, so we don't have good equals. All right, so there are more practice questions with crosses and talking about. All right, epistasis is pretty cool. This is how we can get yellow, black, and brown labs. So if you have a lab at home, it's probably one of those 3 colors, black, chocolate, or yellow. And they show up because they have 2 sets of genes that code for their coat color. And so let's use this mouse example for this, since my other slide looks kind of funky. So in mice, quite like labs, they have 2 sets of genes. One is for coat color, and then one is for pigment production. So in order for this mouse to have the brown, or like wild type, you might see it called, they have to have 1 dominant trait of each. So they have a dominant agouti coat color is what it's called. And then they have 1 dominant pigment color, which creates a dark pigment. So if we lose that pigment, that dominant pigment trait, we get these white albino mice. But if we do the opposite and we lose the coat color and we just have a recessive for the coat color, but we have dominant for the pigment production, we get a lot of pigment, and so we get a black mouse. So epistasis just means that one gene could either like mask or it could even intensify another gene. And this is pretty common with genes. Most of the time, we don't have just one gene that codes for something. Eye color is not one gene. It's two or three, with 2 or 3 different genes code for eye color. And so that's why we can get so many different variations. It's because we have multiple genes that code for it. And then some of them mask each other, which is epistasis. We can also have like environmental factors that help code for genes, but if you take Bio 112, that's where we'll talk about that. We'll talk about like natural selection and evolution. Okay, are there questions about any of these so far? Incomplete dominance, codominance, multiple alleles, X-linked traits, epistasis, pedigrees. Okay, let's just work this question here, and then that'll be it for today. How about that? I don't have a poll question. I was supposed to put a poll question on my last slide, but it's blank. So let's see if we can work through this one here. I'll ask for volunteers. So let's read the question first. Some mice are born with a yellow coat controlled by the gene Y. So it must be like big Y and little y, right? Examine the following crosses. So P0, yellow mouse is crossed with a gray mouse. F1 generation, so their offspring. We have 5 yellow and 4 gray, so it's kind of 50-50, right? P1 generation, so a yellow mouse from this generation is crossed with a yellow mouse from this generation. We get an F2 generation, which is 7 gray, 15 yellow, and 9 of them are dead. Okay, so what is the name of the genetic cross that we just did? I'll put a question mark there, so I'm not sure if it's that. So the name of this cross that we did up here, notice they don't know the genotype. So that's what we call a test cross. When we do a cross between 2 individuals, we don't know their genotype. We're trying to figure out their genotype by their offspring that they have. We're going to do a test cross. Okay. So, which trait is dominant? Which trait is dominant? What do we think? We cross the yellow mouse with the gray mouse. We get kind of 50-50. Let's see. If the trait, they told us the gene is capital Y for yellow coat, correct? So big Y and big Y must be our yellow mouse. And I don't actually know answers for these. I'm working through them just like you. And little y might be the gray mouse. Okay, so that's our hypothesis. We don't know. Let's see. So if we cross them, what we get are all heterozygotes, right? So 1 allele and 1 allele from yellow mouse, 1 allele from gray mouse. And we fertilize, so we're going to get 2 alleles again, 1 from each parent. Heterozygotes, okay? So it said we had 5 yellow mice and 4 gray mice in this first cross. So looking at our Punnett square, this might not be the case for our genotypes, right? We would have, if this was if this was the case, big Y equals yellow, we would have all yellow, and that's not what we have. So I'm going to erase. Start over. All right. Big Y, little y might be our yellow mouse. And then we'll have little for gray. So let's cross them. Big Y, little y allele, little y allele. So one allele from yellow mouse, one allele from gray mouse. So this looks like what we got, right? That looks correct for what they said we had. So 5 would be yellow, 4 would be gray. Great. All right, so what trait is dominant? So I erased our first answer. Oops, I'm trying to do my text. There we go. Test cross, which trait is dominant? Yellow, coat color. So C says, what is the genotype of the yellow mouse in the first cross? So we just figured that out, luckily. We first thought it was homozygous dominant, but obviously we worked it out, didn't work. So we're going to say it's heterozygous. What is the genotype of the gray mouse in the second cross? Ooh, in the second cross. Okay. Let's see. So if we do a second one, we're going to cross we'll get these, we'll get this. So we get big Y, little y, little y, and little y, little y. In a 1 to 2 to 1 ratio. What is the genotype of the gray mouse in the second cross? So if we get a gray mouse in the second cross, which is our baby, right? F2 baby. Little y, little y. And then the last question, why are 9 mice dead in the F2 generation? So what genotype? I'm going to let you guys answer this. What genotype is the lethal genotype? Who thinks they got it? If we have our first cross, my pen is dying. If our first cross, so we had a yellow mouse and a gray mouse, obviously they're living, right? They produced heterozygotes and homozygous recessives, which were fine. They didn't die. But something happened with our F2s. Which of our F2s is the lethal? So yeah, the big Y is the lethal. And we know that because of our other crosses. So we had to do a test cross to figure out what lethal gene was and what our genotypes were.